Okay, folks, let's get started. Happy Friday. 1 week from the exam. Hey. Then that torture is over, right? It's Dad's weekend. How many dads here today? 1, 2, 3. Okay. Whatever they told you about this class is not true. Okay? I want you to remember that. Okay? It's not true. But I think in honor of Dad's weekend, we should do a little something special. Maybe we'll do something different from usual. We'll sing a song. How about that? This is a song that goes out to all those dads out there, those that are here and those that can't be here. And it's to the tune of Danny Boy. You guys have heard this tune before, but it's a different song. Different lyrics anyway. And I hope you'll join with me and sing and show Dad how loud we can sing today, okay? It's called Oh Daddy Dear. So, Daddy dear, this is my biochemistry. The problem's long, the course is really tough. That last exam, it really put the fear in me. I studied lots, I hope it was enough. But let's forget about it while you're visiting. And bong a beer when we get out of class. Then when you're gone, I'll go back to my studying. That Kevin Ahern really, truly is an awesome guy. Okay. I don't want your pity. Thank you. Thank you. Okay. Back to more mundane things like biochemistry. So last time I went through and said an awful lot about signaling, and I'm pretty much done with what I want to say about that. I hope by the end that I convinced you that signaling is a process that has enormous implications. It's when signaling gets screwed up that we can see all kinds of problems happen, and most commonly they happen or manifest themselves in ways that we can relate to, like cancer. Okay? And I talked at the very end about a monoclonal antibody that's used to treat certain types of breast cancer called Herceptin. And Herceptin is, as I said at the very end, a very effective treatment. It's a very non-invasive treatment. The side effects of Herceptin are fairly minimal. And if you have a type of cancer that has an overexpressed it's actually called HER2 receptor, then Herceptin is actually a godsend. There are other things that about signaling. One other thing about signaling that I want to point out, where we use our knowledge of signaling to actually, again, devise a treatment that's very effective for treating certain types of cancer. And this type of cancer is, it seems unusual, but it's actually more common than you might think. But there's a type of cancer called chronic myeloid leukemia, or CML. It was in the news this past week. Former basketball great Kareem Abdul-Jabbar announced that he had chronic myeloid leukemia. And it arises as a result of rearrangement of DNA. Okay? We haven't talked about DNA rearrangements, but DNA rearrangements happen not infrequently. And in this particular type of rearrangement, what happens is that there are two sections of two different chromosomes that recombine and mix and match things. It's not exactly known why and how, but it's a common phenomenon that happens. And when it happens, essentially a new gene is created. Okay? So in this rearrangement, a gene called BCR, which is a normal cellular gene, does its normal cellular things, becomes linked, that is, its DNA sequence becomes joined to a tyrosine kinase known as ABL, ABL. Okay? ABL is an oncogene. It's a proto-oncogene until it gets mutated, in which case it becomes an oncogene. And again, I want to emphasize that proto-oncogene is the unmutated form. The oncogene is the mutated form. What happens when this rearrangement is that the BCR gene becomes linked to the ABL gene, and they form a fusion. So the protein that arises from that is part BCR and part ABL. Okay? The ABL portion of this BCR-ABL fusion still contains a tyrosine protein kinase. Now, what appears, the reason that this fused system appears to cause cancer is because the BCR promoter, which is what determines how much of a protein is made, is kept. So the BCR promoter, which makes a lot of protein, is fused with this BCR-ABL so that the BCR-ABL is made in great abundance. Much greater abundance than ABL by itself would have been. Okay? Since the BCR-ABL fusion contains a tyrosine protein kinase, then what happens is you have an awful lot more of the tyrosine protein kinase from ABL floating around the cell than you do in an unmutated cell. Okay? So we think back to the HER2 case where we had one of the reasons that we had a tumor was we had an awful lot more of the HER receptor, which was interacting with the EGF receptor and causing signaling of things to stimulate cell division that we wouldn't otherwise have. A similar thing is happening here, only here we have an enzyme, a tyrosine protein kinase, that's being made in great abundance, and it too is signaling, telling the cell to divide. So it's a numbers game. This fusion creates a new gene that has a function that you don't want. That's the bad news. That's how you get chronic myeloid leukemia. The good news is that there are drugs that people have designed that target this gene that are very effective. And specifically, they are tyrosine protein kinase inhibitors. Okay? Now, you start thinking about the properties that you want of an anti-cancer drug. One of the things that comes to mind is having a tyrosine protein kinase inhibitor, because tyrosine protein kinases play roles in signaling the cell to divide. The one that is used commonly to treat this gene is called Gleevec, G-L-E-E-V-E-C. It inhibits this enzyme, and it's reasonably specific to this. Okay? It doesn't have an awful lot of other side effects because, again, this is a gene that we see basically only in cancerous cells. You hear people talk about magic bullets. A magic bullet against cancer is a bullet that kills only cancer cells and does not harm regular cells. The reason that you see problems with chemotherapy, with people have their hair falling out and being sick and things like that, is because a lot of normal cells are being killed by the therapy. Okay? When you can design magic bullets that target specific things that are only found in cancer cells, you have a much higher likelihood of having a drug that A, is effective, and B, has fewer side effects. So Gleevec is one such anti-cancer drug, and there are many others. I just want to make you aware of this one. Okay, questions about that? You're a quiet group today. Okay, well, that's what I want to say about signaling. So we're going to turn our attention now to talking about metabolic control. And though it looks like we're behind, we're really not behind, because metabolic control doesn't take an awful long time to go through and talk about. Okay? Metabolic control is another type of control that's existing inside of cells. Okay? And it's related to the things that we've been talking about here. Remember that we need to control metabolism. Metabolism is the sum of the chemical reactions going on inside of a cell. It's a good definition. Metabolism is the sum of all the chemical reactions going on inside of a cell. So metabolic control, therefore, means that we want to be able to control the chemical reactions going inside of cells. And I'll remind you that we have to have a lot of control because we have enzymes that are capable of making thousands or millions of things per second. And if we're not careful about that, we're going to make too much of something. We're going to deplete our resources of something. We may burn up energy of something. And if we don't control that, we're going to be in trouble. So when we talk about metabolism, which is what we're getting ready to talk about for the rest of the term, when we talk about metabolism, we have to think of the importance of controlling those pathways. Without control, the cell will not be able to function in the long term. Okay? All right. So we look at metabolism. Metabolism, to an awful lot of people, looks like a roadmap. All right? Now, that is a schematic that shows you the primary reactions that are found in all cells. Okay? Are you guys looking forward to learning all those? I woke you up there, didn't I? Okay. Well, fortunately, you're not going to learn all of those. I will tell you that as a graduate student, way back in the dark ages, I actually at one time knew every one of those things that was on there. Couldn't do it today, but that's a not unreasonable thing for a graduate student, for example, to be able to. Would the 550 students in here like to do that? Are you speaking for yourself, or are you speaking for the group here, Laurie? So, okay. All right. So, in any event, it looks like a roadmap. Okay? And there's a connectedness to it. When we think about metabolism, we probably never thought about it before, but every reaction is connected to other reactions, just like highways intersect and connect with each other there. Okay? When we talk about glycolysis, and I'm going to show you a schematic of glycolysis in a second, when we talk about glycolysis, what we see is that we have a molecule, glucose, that gets converted into something else, and then that something else gets converted into something else. So the product of one reaction becomes the reactant of another reaction. There's a connectedness. A goes to B, B goes to C, C goes to D. Okay? We can't escape that fact, and that's a linear progression. Okay? So we see a linear progression if we start here at glycolysis and we go down, 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 down. Okay? But we also see that there's side paths. C in this pathway here for glycolysis also can go over here to another pathway and make something else. Okay? Now, if it seems like that's extraordinarily complicated, you are exactly right. Okay? It is extraordinarily complicated. And I don't expect that you're going to understand the subtleties of that, but I do think that you should understand major intersections, just like you would understand major intersections on the way to Portland or on the way to San Francisco. Off-ramps, on-ramps, we have these sorts of things in metabolism, and they're important for us to understand. Okay. Let's look for a moment at glycolysis. Glycolysis is the study of glucose metabolism. That's where it gets its name. Glycol means sugar. Lysis means break. So glycolysis is a pathway that's involved in the breakdown of sugars. We will see after we talk about glycolysis that other sugars besides glucose can be broken down in glycolysis, so the name is an appropriate one. It's a way of breaking down sugars. Glycolysis, as this schematic shows us, has 10 steps. 10 enzymes, 10 steps. A goes to B, goes to C, goes to D, goes to E, F, G, H, I, J. Okay, we have 10 steps, 10 enzymes. Every step catalyzed by an enzyme. The product of one is, in fact, the substrate for the next one. We look at glycolysis and we think about metabolic pathways, and one of the things you're going to hear me say, and you're going to hear a lot of people say, but it's not really right, is that we talk about a beginning and an end for a metabolic pathway. It's not really appropriate for us to talk about that any more than it's appropriate for us to talk about the beginning or end of I-5, for example. Right? I suppose if we went all the way up to Canada, we actually do have a beginning. But any other place in I-5, we don't really have a beginning. It's a continuous path that goes through there. Metabolic pathways do not have beginnings and they do not have ends. They're man-made inventions. So when I talk about glycolysis, what I'm doing is I'm picking out from that roadmap I just showed you 10 connected reactions, but they're occurring in a soup where any of those intermediates can go off into something else. That's a very important concept to keep in mind about metabolism. Those intermediates are products and substrates of other pathways. So glycolysis intermediates can go make something else, and there are other pathways that will feed intermediates into glycolysis. Okay, so we keep that in mind. This shows what we like to think of as the beginning and the end of glycolysis. The beginning is glucose. The end is pyruvate. But in fact, pyruvate can go off and make other compounds. We'll talk about those later. Okay? So glycolysis is a definition, as it were. It's a definition of 10 steps of breaking down sugars. That definition, that pathway, is in the midst of a roadmap of other things that are going on. I always want you to keep that roadmap in mind. Okay, so when we get to pyruvate, we have really two fates in our cells for pyruvate. It can go two different directions, and we start to see that metabolic pathways have conditional directions. They can go one way or they can go another way depending upon conditions. In this case, if your cells are running out of oxygen, if you are exercising heavily, your muscles are using glucose faster than the body can provide oxygen to allow its full oxidation. When that happens, you run out of oxygen, you go anaerobic. And when you go anaerobic, the body has to do something with that pyruvate. It makes lactate out of it. When you have plenty of oxygen, that's not a limitation. The body takes that pyruvate and says, oh, we'll make acetyl-CoA out of this because I can burn this in the citric acid cycle and make a lot of ATP. So we see conditional pathways going here versus there depending upon conditions. Those conditions, in many, many cases, as we shall see, relate to concentrations. Okay? So I'm going to talk, and understanding concentrations is important because concentrations affect the Gibbs Free Energy, and the Gibbs Free Energy determines directions of a reaction and the favorability of a reaction. So it's not surprising if I had an awful lot of lactate piled up inside of cells, this pathway may not be very favorable. Okay? So we could imagine that there's going to be things that are going to change inside of cells as a result of conditions, and those are going to affect metabolic pathways. Okay. ATP, your book likes to describe, and I think it's appropriate, is a metabolic currency. Okay? Currency is something that you can spend. You can take dollars and you can go anywhere in this country and spend dollars. It doesn't matter where you take them, you're going to be able to spend dollars. In a few foreign countries, you can actually take dollars and spend them there as well. It's not a universal currency, but it is a common currency. Okay? ATP is similar in the cell. ATP is a high energy source. What we're going to see in talking about metabolism is that we have to consider reactions that produce energy and reactions that need energy. It's not surprising that when we go building something, it takes more energy than it does to break something down. If we need energy, our energy source is commonly, not always, but commonly going to be ATP. ATP, the triphosphate, of course you know, is what I've referred to in class as the gasoline of the cell. It's what makes things go. Okay? Now, what we're going to find as we talk about metabolism is frequently confusing to students. And the confusion is that they want to see that energy of ATP sort of going pow, like gasoline in a piston, and as a result of that, something happens. And that's not what happens with ATP. ATP, the energy of ATP, is used in what are called coupled reactions. Coupled reactions. I'm going to explain that to you in a second. So a coupled reaction is a reaction that takes a high energy compound and couples the release of energy with it to an energy needing reaction. Now, that's kind of vague. I'll give you an example in a second. But a coupled reaction takes a high energy compound and couples the release of energy from it to an energy that requires to a reaction that requires energy. Okay. Well, in practical terms, what does that mean? Well, I'll give you an example, and I'll talk about it later when I talk about glycolysis. The very first step of glycolysis is the conversion of glucose to glucose 6-phosphate. Okay? There's a couple ways we can imagine this reaction could occur. One way is we could take glucose. We could take the enzyme. The enzyme is known as hexokinase, which you don't need to know right now. We'll talk about it later. Take the enzyme, take glucose, take some phosphate, mix them together, and look to see how much glucose 6-phosphate we make, because the phosphate and the glucose are going to come together in the enzyme and make glucose 6-phosphate. Okay, we can do that. And if we do that and we look inside the cell, what we discover is that very little glucose 6-phosphate is made. Very, very little. Why? Because this reaction requires energy. Okay? Then you say, oh, then we have to just go burn some ATP and it goes. Well, it's not quite that simple. Okay? Instead, the enzyme has a binding site for glucose and it has a binding site for ATP. There's really two reactions that's going on inside of this enzyme. One enzyme is the transfer of phosphate to glucose to make glucose 6-phosphate, and that phosphate is coming from ATP. The second thing that's happened is ATP is giving up that energy by breaking that phosphate bond and becoming ADP. So I've coupled the breakdown of ATP to the synthesis of glucose 6-phosphate. I made them both occur in the enzyme at the same time. I didn't do that. The enzyme did it. The enzyme had the binding sites for it. There wasn't any great effort or brain part on my part. Okay? So, when we look at metabolism over and over, we discover that that is in fact the way that energy is realized to make unfavorable reactions move forwards. And you say, well, chemically at the level of the enzyme, what in the heck is happening? Okay? That energy is released. Does the enzyme just grab that energy and do something with it? Okay? It doesn't. It doesn't. We can imagine that we've got an enzyme, that we've got triphosphate there, and so what actually happens with this enzyme, I've sort of schematically shown this to you before, is I have a binding site up here for glucose, I have a binding site down here for ATP, and the binding of the two causes a structural change in the enzymes such that they come closer together to each other, and that putting them close together in each other puts the phosphate down here close to the glucose place where it goes up here, and the phosphate moves over. Where's the energy? How does the energy play into that? Okay? Well, the energy plays into it when you break that phosphate bond on the ATP, which the enzyme helps to do. When you break that phosphate bond, you actually are doing something like, I don't know, I guess when I was a kid, my dad wasn't very good about lighting fires. And so you have this experience where, oh, we don't need a charcoal lighter, right? Little gasoline, take care of it, right? You're lighting the charcoal. Mother's going, no, 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 right? Okay. A little bit of gasoline on there and kaboom, right? Singe the eyebrows, you get all this. That kaboom, okay, caused a physical change to happen. Okay? The physical change inside the enzyme has to be very small, as you know. A slight structural change can now happen as a result of energy that we put in, and that's what happens in ATP. In the enzyme that's cleaved ATP. A slight structural change has happened that's finally pushed these guys closer to each other such that the phosphate now ends up being on that glucose. So the energy that is released results in changes inside of the enzyme, and those changes inside the enzyme now make things happen that wouldn't happen very well otherwise. Over and over and over now, you've seen that slight changes in a protein have tremendous effects on what the protein does. Here is a direct energy component that we haven't seen before. Yes, Laurie. That's correct. Binding of ATP and glucose causes a structural change. The breaking of the phosphate off there causes the final structural change that allows that reaction to occur. Okay? Okay, so that's a simple schematic about how an enzyme does what it does and how an enzyme uses energy. Talking about energy, we need to think about that in a little bit more depth. Your TAs have been talking to you in recitation about the Gibbs Free Energy, and they've been working some problems for you. And I want to go through and say a few words about Gibbs Free Energy and get you thinking about problems. And I think most people find that Gibbs Free Energy problems are not as bad as Henderson-Hasselbalch because they've kind of gotten used to Henderson-Hasselbalch. Henderson-Hasselbalch taught us, of course, that the pH equals pKa plus the log of salt over acid. We had a variable term, the pH. We had a constant term, the pKa. We had variable terms of salt and acid. As we change salt and acid, pH will change. pKa doesn't change, right? We look at the Gibbs Free Energy change equation, and we see that delta G equals delta G0 prime plus RT times natural log of products over reactants. Okay? There's the equation right there. It parallels. It absolutely parallels the Henderson-Hasselbalch equation. We have a variable, delta G. We have a constant, delta G0 prime, for a given reaction. We change products and reactants, we change this ratio, and as we change that ratio, we make it more positive or negative, depending upon whether we add more products or reactants. If I add more products, I'm going to make this log term, and that's just a natural log. It's still a log term. It has all the same rules as the base 10 log. Okay? If I increase the amount of products, I'm going to increase the value of that ratio. I'm going to make that log term more positive. And when I make that log term more positive, I make the overall delta G more positive. Conversely, if I increase the amount of reactants, I make this ratio be smaller. Therefore, the natural log term becomes more negative, and the overall delta G becomes more negative. Now, we've talked about before, and you've seen it in your freshman chemistry classes, that delta G is a very, very useful parameter because it tells us the direction of a reaction. When delta G is negative, the reaction goes forwards. When the delta G is positive, the reaction goes backwards. And when the delta G is equal to 0, we're at equilibrium. That's not any different than Henderson-Hasselbalch, which told us that pH equals pKa when salt equals acid. Right? Okay? So we start to see then how this equation can be helpful to us to understand directions of metabolic reactions. We're going to see that the breakdown of glucose in glycolysis goes in the direction that I started before, showing it down. We oftentimes think of it going down. We start with glucose at the top, and we end up with pyruvate at the bottom. That's great if we're breaking down glucose. What if we want to make glucose? We can make glucose in our bodies. Our liver does it all the time, starting with pyruvate. Guess what it does? Most of the reactions of glycolysis are simply reversed. The question then arises, are we going forwards or backwards? Are we going in the breakdown or the synthesis pathway at any given time? And that's going to change depending upon how much of the materials that we have there. If we have a great abundance of pyruvate, which is the end product, you could predict that that's probably going to favor the backwards reaction. Why? If I increase products, what does that tell me? It tells me that the log term becomes more positive. The backwards reaction is favored. Instead of breaking down, we're going to be making. Okay? It's a simple example, but that's a very effective example about how the ratio of these help us and tell us something. The delta G0 prime, as I've told you before, is something you're going to confuse with delta G because they sound alike. So just call the delta G0 prime Clarence. Doesn't sound anything like delta G. Delta G equals Clarence plus RT times natural log of products over reactants. It's so simple when you call it Clarence, right? Clarence is a constant for a given reaction. Are there any Clarences in here? I don't want to, favor one student over another, Okay. All right. It's just a constant for a given reaction, just like pKa was a constant for a given buffer system, right? Don't mix the two. Delta G0 prime turns out to be the same as delta G under certain conditions. Just like pH equaled pKa under certain conditions, delta G equals delta G0 prime under certain conditions, and those conditions include what are called standard conditions. Standard conditions involve 1 molar product, 1 molar reactant, equal amounts of the two. When these two are equal, that ratio is 1, and what's the natural log of 1? 0. Exactly the same as the Henderson-Hasselbalch equation. At equal concentrations under standard conditions, delta G0 prime equals delta G. The standard Gibbs free energy, is equal to the Gibbs free energy when we're under standard conditions. But most of the time, we're not under standard conditions. Standard conditions happen in the laboratory where we put the chemicals together and we make those things. We make that situation. That's when that we have standard conditions. Delta G0 prime tells me the direction of the reaction only, underline this, only under standard conditions. It doesn't tell us any other time. If the delta G0 prime is negative under standard conditions, you may assume that the reaction will go forwards. If the delta G0 prime is negative and it's not under standard conditions, the safest thing for you to put on the exam is, we don't know. We have no way of knowing. Why? Let's say this term is negative. Is it possible for delta G to be positive? Absolutely. What if we had this term be very, very positive? Very big. This positive outweighs this negative. The delta G could be positive. We can't tell if we're not at standard conditions. Everybody understand that? A few nervous, yes, yes. How many of you have worked the problems? How many of you are confused by the problems? You can be honest, okay? All right. If you're having problems with the problems, see your TA, come see me. I'm happy to step through them and work through them with you. And as I said, most students, I think, find that understanding that concept is not nearly as complicated as what they felt the first time they did Henderson-Hasselbalch, and it's because of the parallels. Okay, well, ratio of products and reactants we're going to see is very, very important. Ratio of products and reactants is going to determine for us if a reaction goes forwards or a reaction goes backwards. And it's important to remember that virtually every reaction is reversible. One of the things that you'll hear, and I'll probably slip into saying this as well, so I'll make you aware of it, but you'll hear people talk about the delta G0 prime for a reaction as really helping a reaction to go forwards or backwards. Forwards or backwards, okay? Let me give you an example. If we look at the last reaction of glycolysis, it's a conversion of a molecule called PEP into pyruvate. And the delta G0 prime for that reaction is something like minus 37 kilojoules per mole. It may not seem like a big number, but that's a very, very large negative number. Very, very large negative number for this equation. If I had that reaction, does that mean that that reaction will always go forwards? No. What's it going to take for the reaction to go backwards? It's going to take this term to be positive, and for this term to be positive, the ratio of B to A is going to have to be enormous. If I'm at equilibrium conditions, or I shouldn't say equilibrium conditions, if I'm at equal concentrations of product and reactant, which direction is the reaction going to go? Forwards. Okay? So keep that in mind. When people talk about this determining or helping or influencing the direction of a reaction, it is an influence, but it's not the determinant. Most commonly, people, including myself, will compare the delta G0 prime values of one reaction versus another reaction. We'll see that in glycolysis, many reactions have delta G0 primes close to 0. What does that mean? It means that if we start with equal concentrations of the two, we don't get much direction one way or the other. If we start with a delta G0 prime that's very positive, and there's one reaction in glycolysis that is very positive, it means that when we take equal concentrations of the two, the main direction of the reaction is going to be backwards. Conversely, like the one I've already explained to you, if I have a delta G0 prime for a reaction that's very negative, if I start with equal concentrations of them, it's going to be very favorable forwards. Okay? So that gives you a feel, I hope, for what those terms mean and how those terms are useful. Please work through the problems. Yes, Marcia? Yeah, our question is temperature constant? One of the things I don't talk much about is temperature in this equation, because temperature is a variable, actually. All right? For biological systems, temperature is pretty much a constant. Your cells don't change much from one second to the next in terms of their temperature. Or even an organism that lives in an extreme environment, from one second to the next, the temperature doesn't change much. So we can generally think of temperature as behaving something like a constant in a biological system. And you may assume, for our purposes, we'll say 300 degrees Kelvin And I'll usually give you that on a problem or an exam. If I don't, you may assume that. Okay? Okay, so that's what's up with delta G0 prime. Now, molecules and energy are important things to think about because we will see as we get going through that there are molecules in the cell that are full of energy. ATP is one of them. You've already seen that. Full of energy. That energy is useful for something. It's useful for making things. Here's glycerol 3-phosphate. Glycerol 3-phosphate is a molecule that has a phosphate on it. And as I've told you in class before, putting phosphates onto things generally makes them, have a higher energy. Glycerol 3-phosphate, I have hiccups. Glycerol 3-phosphate has more energy than glycerol by itself. If I want to make that, it means I've got to put energy into making it. Just like if I want to build a skyscraper, I'm going to have to put some energy into constructing that thing. Similarly, I have to put energy into constructing this thing. This is the skyscraper. Well, glycerol 3-phosphate has some energy in it. There are molecules that have more energy than glycerol 3-phosphate. Okay? Here are some molecules we'll talk about a little bit. Two of them occur in glycolysis, phosphoenol pyruvate. Okay? It's got a phosphate, it's got a negative charge, and it's got a double bond. And this guy is absolutely loaded with energy. Here's another one we find in glycolysis. 1,3-bisphosphoglycerate, or 1,3-BPG, which sounds like something we've talked about before. Okay? And it's actually related to it. This guy has two phosphates, one on this end, one on this end. Okay? These phosphates repel the heck out of each other. They want to get away from each other. That's like a spring that is pushed together. It wants to spring out. These phosphates want to spring away. That means there's high energy in there. Potential energy that can be used for something. Okay? Well, I want to spend about 5 minutes talking about a compound that you guys hear a lot about. Okay? Athletes hear a lot about this compound. Creatine. How many people have heard of creatine? How many people think, wow, this is the way to go? You get creatine, man, and you got more muscle power, and you got more energy, and I can be an Olympic athlete, right? We all hear these sort of things, right? How many people believe that? Now, nobody's going to raise their hand now. No, I'm not going to do it. No. Well, there we go. Okay, there's one brave soul. Good, good. He believes this. This is good. Okay, so what I'm going to do is I'm going to just sort of shoot some of that notion down. Not completely, but I'm going to shoot some of that notion down for you. Okay? Creatine. Well, creatine is a really good thing. This is creatine phosphate. It's a really good thing for us to think about relative to metabolism and relative specifically to muscles and what muscles do. Creatine is found in muscles. And it turns out that creatine is a compound that is made in a reaction that looks like this. Okay? Here's the reaction. Creatine plus ATP. And I use that little squiggly thing there to mean both directions for a reaction. Goes to creatine phosphate plus ADP. Okay? Now, what happens with this guy is that the cell uses this as a way of storing energy. So muscle cells have to be able to have a lot of energy released very quickly because if you have to sprint or you have to escape that grizzly bear I always talk about, you have to be able to burn a lot of energy quickly. So when you're sitting around the house, drinking beer, watching the tube, eating pizza, your body is doing you a favor, so it thinks, because it's saying, well, you're not burning up all this ATP. The concentration of ATP in your muscle cells is getting pretty high. We could let that sit there and do nothing, and it wouldn't do us a lot of good because ATP after a point is going to be a problem. So we should do something with that energy. We should store that energy in some way. And so as the ATP concentration is high, the phosphate and ATP is transferred to creatine to make creatine phosphate plus ADP. Okay, this is an enzyme. The enzyme that catalyzes this is known as creatine kinase. It's abundant in muscle cells. Now, creatine phosphate has a lot more energy than ATP does. I'm going to say that again. It has a lot more energy than ATP does. How in the world can we make a molecule that has more than ATP does by starting with ATP? Hold on here. How do we do that? Based on the Gibbs equation, what would we need to do?
But are you already coupling it right with ATP going to ADP as part of it?
Okay, so he's got the coupling reaction, but what I'm telling you is the coupling reaction alone doesn't provide enough energy to make a higher energy molecule. Is this an impossible thing? It must not be. He put it on the board, so yeah.
High concentration of ATP compared to ATP.
Okay, so very good. Remember what the equation tells us. Concentration of products over reactants. If we want to drive a reaction forward, we want delta G to be negative. How do we make delta G be negative? We have to lower this ratio. The smaller this ratio is, the more negative this term is, and the more likely it's going to go forwards. And how do we lower that ratio? By increasing the amount of reactant. The more ATP, remember I said we're sitting around drinking beer, eating pizza, and watching the tube. We're not burning up ATP. Our ATP concentrations in our muscles are high, and I said we wanted to do something with that ATP. Here's what we're doing with it. This reaction becomes favorable as the ATP concentrations go high. Very cool. ATP concentrations are high. We're storing energy for that moment 5 minutes from now when somebody comes running screaming, the house is on fire. The house is on fire means that we had better have some energy to get our tails out of that house, okay, before the fire completely engulfs us. Okay? So we jump up and we happen to live in a very big house. Maybe we're in a fraternity or a sorority house. And just jumping doesn't get us out of the house. We have to get up and we have to run. Maybe we have to go to our room and grab all of our belongings and get them out of there. We've got a lot of panic. We've got a lot of excitement. We've got a lot of things we've got to haul out of there. Maybe our drunken roommate that we're grabbing by the ankle and pulling them out of the house. We have to do this. Okay? Do you guys have a lot of drunken roommates? I see a lot of smiles when I said that one. You do? Okay. Have you had to do this?
No.
You can tell them at the molecular level, or her, how you saved her life. Okay? All right. When I start that process, what happens? Well, my muscle cells have some ATP sitting around. And ATP is necessary for muscular contraction, so I grab and I pull, and the very first thing that happens is my ATP levels fall. My heart hasn't recognized yet that this is an emergency. It hasn't started beating faster, so it's delivering more oxygen and more nutrients. It's not bringing anything to those muscle cells. All those muscle cells have is what they happen to have in them. And since they've already been using ATP to make creatine phosphate, they don't even have an awful lot of ATP. That initial jump burns what little ATP that they have. Uh-oh. This is a problem. The same thing happens when you go to run a 100-yard dash. The first few steps that you take, you burn up what ATP the muscles have. If you didn't have a backup supply, you are going to fall flat on your face. You're not going to rescue the roommate. Look at this reaction. What happens when ATP concentrations fall? Which direction does the reaction go? It goes backwards. And what do we start making? ATP. Isn't this cool? This is wonderful. Okay? We've made ATP, and we've used our battery, as it were. Our battery backup is creatine phosphate. Now people say, now I see why. If I take creatine, I'm going to be a super athlete. Right? I'm going to be a super athlete because I'm going to take a lot of creatine, and I'm going to make this thing go, right? All right, you super athletes, I want you to be super brainy athletes. Okay, let's think about this. Let's say I'm getting ready to go run my 100-yard dash. The house isn't on fire, but I'm going to run like it is when I'm in this 100-yard dash. Creatine's good. Let's go have some good creatine breakfast before we go out on our run, right? Great, let's go have some creatine. We're going to do all this here, and we're going to have plenty of creatine. Now let's go take off on that run. What's going to happen? Well, our ATP concentrations are going to fall, but uh-oh. What else do I have in that equation? Creatine. What's going to stop the reaction from coming backwards? Creatine. Creatine is a reactant. I just shot myself in the foot before I even started my race. Now I get the question, but what if you took creatine phosphate? You know, everybody wants to win the lottery. Everybody wants to cheat. Everybody wants to have a way to get it there. Okay? The answer is you probably couldn't take creatine phosphate because as soon as it hit your stomach, it would probably just break down into creatine, and now you're right back where you started. Do you want to take a chance of that as an aspiring Olympic star? No. Okay? Now, there are clinical studies, and I'm painting a sort of an odd example to you, where actually there are some performance things with creatine that may have some benefit. Okay? I certainly don't recommend taking things to increase athletic performance. And the reason I don't do that is there are several reasons I don't, but one of the reasons that I don't is you really don't know what the long-term effects of doing something like this is. Okay? You have no idea what the long-term effects of doing something like this is. And what you don't want to do is muck with something. If we look at professional athletes and we look at the life expectancies of professional athletes, they're less than that of the regular population. Okay? That isn't to say that we shouldn't exercise and we shouldn't be athletic and we shouldn't do things and we shouldn't have sports teams, but I think that we should be careful about what we do to get there. And that's my point. Okay, let's see. That's my sermon for the day. Let me finish up with a couple other things and we will be done. Okay. Energy. This is all about energy. Energy is an important consideration because we need energy to be alive. And to be alive, we have to burn things to get energy. How do we get ATP? By burning things like sugars. We literally burn them. We oxidize them. Okay? What we see in our body is that we have reactions that make ATP, and this is shown in blue going to the left. These are oxidation reactions that produce energy that is then captured in the form of ATP. And we do this because ATP is necessary to make things. We have to be able to make proteins. We have to be able to make carbohydrates. We have to be able to make fats. We have to do all of these things. And making things takes energy. Energy in the form of ATP. So we have this sort of perpetual cycle. We have to oxidize to make ATP. We use that ATP to make things that are important to us. This is an essential part of what living is all about. Okay. Oxidation. Okay. Oxidation, I've said it before, I'll say it again here, involves the loss of electrons. If we compare the oxidation state of something like this, and this is something that you should have all learned in organic chemistry, this is the lowest oxidation state. We go from the lowest oxidation state to the highest oxidation state. What happens is at each step, we release energy. Lighting a match, lighting a fire, we are oxidizing. We're going from a lower energy state to a higher energy state, a higher oxidation energy state. Okay? We're doing that. We're burning this. All right? Methane has this much inherent energy in it that can be released upon oxidation. Carbon dioxide has virtually nothing in it that can be released upon oxidation because it's completely oxidized. It's at the highest oxidation state. We see varying levels in between. Here's a hydroxyl. Here's an aldehyde. Here's an acid. Here's carbon dioxide. Okay? In general, things that are acids are, not in general, things that are acids are at a higher oxidation state than aldehydes or alcohols or just plain old carbon molecules. Okay. We see in metabolic processes that there are oxidations that happen, and this is what I'm going to finish with here. Okay? Oxidation happens inside of cells, not, underline this, not in the same way that oxidation happens out in the real world. In the real world, I light a match, I light a fire, I burn, I get a lot of heat off of that. If I had those reactions going on inside of me, my temperature would be a lot higher than it is. You talk about a fever, you'd have a fever. Biological oxidations are much more controlled, and they're controlled using two things, enzymes and electron carriers. Electron carriers are essential because they allow tiny oxidative steps to occur, and those tiny oxidative steps don't overheat the system. If we heat up a cell too much, what's going to happen to our proteins? Denature. We're dead, right? We want to have controlled oxidation such that things are not going to heat up too much. Okay, that's a good place to stop. I'll talk more about that next time. How you doing, Jessica?
I'm doing all right. How are you?
Good. What's up?
I was wondering if... So I was just wondering, when you're talking about how, when you're talking about the reactions with ATP to produce energy, is that always going to occur with an enzyme as a medium for that?
It will always occur with an enzyme, yes.
Okay.
Yes. So
Okay.
coupling will always occur on an enzyme.
Okay. And then I was also just wondering, what you're talking about for what causes CML?
Yes. No, it's not. It's not.
Okay.
It's not. Is that what your father has?
Yeah, my dad has CML.
Okay. Yeah.
All right. Thank you.
Yep. You bet. Take care. Hey, Andy.